Bismillah ar-Rahman ar-Rahim. The cytoskeleton is a complex network of protein filaments that form a supportive scaffolding within the cell. In the waiting room, we have a boy with Duchenne muscle dystrophy. Study of cytoskeleton may help in understanding the cytoskeleton protein dysfunction in this boy. Plasma membrane is the outer boundary of the cell. Organelles such as endoplasmic reticulum, ribosomes, and mitochondria reside within cytoskeleton framework. Microtubules organize the cytoplasm, often interacting with organelles to produce organelle movement. Three types of filaments are present in cytoskeleton. Out of these, microtubules and actin are composed of globular subunits, whereas intermediate filaments are fibrous proteins with alpha helical structure. Within a cell, actin is often localized to regions near plasma membrane called the cell cortex. In cytoplasm of the non-muscle cells, actin regulates the physiological state of the cytosol, which is the liquid portion of the cytoplasm without organelles. Within the nucleus, actin filaments are important for chromatin and play part in regulation of gene expression through epigenetics. Actin filaments are supported by actin binding proteins, which include spectrin, which forms flexible network beneath cell membranes on cytosolic side, and dystrophin, which attaches actin filaments to basal lamina and provides tensile strength to the muscle. So the boy in the waiting room is having muscular dystrophy in which the dystrophin protein is defective, and here we can see that the function of the dystrophin protein is that it attaches the actin filaments to basal lamina, providing tensile strength to the muscle. Myosin is also an actin binding protein. Myosin 1 and 5 help to move materials on tracts formed by the actin, whereas myosin 2 helps actin filaments in cell cortex to maintain cell shape. This is an animated representation of the role of actin and myosin in cytokinesis or pinching off of cytoplasm when myosin 2 slides actin filaments over each other to mediate local contractions. Microfilaments are formed by polymerization of F-actin from a by polymerization of F-actin to form a tension cable, which not only circles inner portion of epithelial cells and regulates their shape, it also causes contraction in wound healing. Furthermore, during division of cytoplasm in dividing cells, actin-myosin bundles form contractile ring, which helps to pinch out the cytoplasm, leading to formation of two daughter cells after nuclear division. Actin is made up of globular or spherical protein subunits. These subunits are called G-actin monomers. These bind to ATP, which helps them to join to form F-actin polymers. After their joining, ATP is converted to ADP. So these are the F-actin polymers which are being formed by the polymerization of G-actin monomers. F-actin polymers have a plus end to which more actin subunits are continuously added and a minus end from which G-actin subunits fall off one at a time. As one subunit is added to the plus end, two strands of G-actin subunits wind around each other to form mature actin strands. Formation of F-actin polymers consists of a lag phase when three G-actin subunits with bound ATP join to form a nucleation site. Then new ATP bound G-actin subunits are added to it. This forms the plus end. ATP is hydrolyzed to ADP, and this is the polymerization phase as the new subunits are being added. Finally, a steady state is reached when the rate of addition at plus end equals the rate of subtraction at the minus end. That's the schematic representation of steady state. If the blue colored G-actin subunit is followed, it can be seen that it appears to be walking in the field until it ultimately falls down from the minus end. So that is the G-actin polymer, and as new polymers are being added, new monomers are being added, it is showing displacement along the F-actin polymer. Now you can see that it is almost in the middle of the polymer, and now it is reaching its minus end, and now finally it is going to fall down. So after falling down, the G-actin subunit again reaches the plus end with new ATP attached and continues its journey again. That is why this motion is called treadmilling. The gel state of the cytoplasm is shown in the lower portion of this slide, where the F-actin polymers are joined together by bundling proteins and cross-linking proteins. The upper portion of the slide shows sol or soluble state of the cytoplasm, which is the result of twisting and breakage of F-actin filaments by coffulin protein and fragmentation or breakdown of F-actin filaments by gelsolin protein. Not only that, sequestering proteins remove G-actin subunits from F-actin polymers. Thus, polymerization is inhibited by sequestering proteins and gelsolin, creating the soluble or sol state of the cytoplasm, whereas the polymerization leads to the gel state of the cytoplasm. The inner or cytosolic side of red blood cell membrane is shown here. Actin binding protein spectrin network is shown by brown color and actin filaments by purple. Presence of spectrin helps to form biconcave shape of RBCs. Another protein and chirin facilitates the interaction between actin and spectrin. Here it is shown that if spectrin is deficient, the RBCs cannot achieve biconcave shape. They become spherical and can break up during passage through the small capillaries. While actin polymerization helps to propel a cell forward, the contraction is needed to pull the plasma membrane of the lagging end, and in this way the cells can move. Next are the intermediate filaments. Intermediate filaments are fibrous with alpha helical structure. These join in sets of 2 to form dimers or in sets of 4 to form tetramers with antiparallel orientation, where carboxyl terminal of one alpha helical filament is opposite amino terminal of the other filament. So that's a diagrammatic representation of intermediate filaments. Type 1 and 2 intermediate filaments form a network from nucleus to the plasma membrane. Type 3 intermediate filaments provide support and structure to the cell. Type 4 include neurofilaments which protect cell against mechanical stress. Type 5 form nuclear lamina, whereas type 6 is involved in growth of nerve cells. This is an illustration of the structure of dimers of intermediate filaments forming coiled coils. Now coming to the third type of the filaments, which are the microtubules. Linear chains of alpha and beta tubulin heterodimers self-assemble into protofilaments. A ring of 13 tubulin molecules embedded in centrosome forms the nucleation site onto which microtubules are built. The beta tubulin end appears to be oriented away from the centrosome. Microtubules continuously switch back and forth between growing and shrinking phases. Therefore, they are said to have dynamic instability. Here it can be seen that 13 protofilaments form the outer wall of cylindrical microtubule structure. Growing or plus end of the microtubule has GTP cap and is directed away from the centrosome. It grows in search of organelles. When addition of GTP bound tubulins to protofilaments slows down, hydrolysis of GTP to GTP becomes more as compared to new GTP flowing in. The GTP cap is lost. New tubulin heterodimers cannot bind GDP bound heterodimers, so not only its growth is stopped, it becomes unstable and protofilaments peel back and the microtubule is disassembled. It may disappear or it may start growing again. Microtubules at poles pull the spindle apart. Kinetochore microtubules attach to kinetochore structures of the duplicated chromosomes and astral microtubules that radiate out position the spindle during the cell division. Centrosomes organize the microtubules and regulate their number, location, and orientation. In cilia and flagella, 9 specialized pairs of microtubules form a ring and surround two additional microtubules. Dynein is a microtubule binding protein that generates the sliding forces between microtubules in flagella. So dynein and kinesin are microtubule motor proteins. They have ATP binding heads and tails that bind stably with their intracellular cargo. Dyneins move along microtubules towards the centrosome or the negative end of the microtubules. Kinesins travel along microtubules away from the centrosome towards the positive end. Both types of proteins hydrolyze ATP for their own movement. So that's a kinesin transporting mitochondria. To summarize, the cytoskeleton is a complex of protein filaments found throughout the interior of the cell. Three principal types of protein filaments of the cytoskeleton are actin filaments, microtubules, and intermediate filaments. Accessory proteins bind to and regulate the function of the cytoskeletal proteins. Functions of actin in non-muscle cells include regulation of physical state of the cytosol, cell movement, and formation of contractile rings in cell division. Actin polymerization occurs by addition of G-actin monomers onto F-actin polymers in an ATP-dependent manner. Treadmilling is the dynamic process of addition of new G-actin monomers to a growing chain, followed by its displacement along the F-actin polymer and its removal from the chain as new G-actin monomers join the chain at the plus end. Intermediate filaments are stable rope-like structures that provide strength and support. Microtubules are involved in chromosomal movements during nuclear division, in formation of cilia and flagella, and intracellular transport. Composed of tubulin heterodimers, microtubules have a dynamic instability and continue to assemble and disassemble according to the needs of the cell.